Food insecurity is a global issue, and in recent years, it's been on the rise. It is the United Nations' second developmental goal. Food insecurity is the lack of regular access to enough safe and nutritious food for normal growth and development in an active and healthy life. Food insecurity affects people from all walks of life. Pregnant women need twice as much iron as non-pregnant women. However, the odds of iron deficiency in food insecure pregnant women were nearly 3 times higher compared for food insecure women. Food insecure pregnant women had a 2.2 times higher rate of mental health issues compared to food secure women. Likewise, the odds of behavioral problems among children with food insecure mothers were 2.1 times higher than among children with food secure mothers. Black and Hispanic households have twice the rate of food insecurity compared to white households, and only 40% of seniors that qualify for food stamps have signed up. Decades of research has demonstrated a strong correlation between food insecurity and poor physiological and mental health outcomes, such as obesity, diabetes, or hypertension. In the U.S., we spend $77.5 billion in additional healthcare expenditures due to food insecurity every year. The Big Bend area is not immune to this problem. The Big Bend contains 6 of the 10 most food insecure counties in Florida, and since the pandemic, our food insecurity rate increased from 14 to 30%. At least 150,000 members of our community are experiencing food insecurity. To learn more about the causes of food insecurity in our community, we spoke with the CEO of the largest food bank in the Big Bend area, Monique Van Pelt. The asset-limited, income-constrained, and employed are the ALICE report highlights how many people are living above the poverty level, but their salaries don't cover their cost of living. In the Big Bend, 53.6% of households are living paycheck to paycheck, leaving many households one surprise expenditure or economic downturn away from a financial crisis. This can result in trade-offs, forcing people to choose between food or housing, transportation, medical care, or utilities. Another driver of food insecurity are food deserts, or areas that have limited access to healthy and affordable foods. Food deserts are communities that suffer from chronic food insecurity because they're located miles away from a grocery store or the residents can't afford healthier choices. To create a more sustainable impact in our community, our team worked with local food pantries and food banks who are leading the effort to combat food insecurity. After speaking with Grace Mission, one of the largest food pantries in Tallahassee, we learned that balanced nutrition is a large problem. Our local food pantries are asking for more fresh produce to increase the nutritional intake of their clients, but preserving fresh produce is difficult. Food pantries that are open once a week can't reliably get fresh produce out to their community. Fresh produce donations received by the food bank only last a few days. Between the food banks receiving fresh produce and getting it out to the community, there exist 3 short windows that the food has to go through. 1. Food banks delivering fresh produce to food pantries. 2. Food pantries holding the fresh produce until clients receive it. And 3. Clients receiving the food and when they use it. At any point in this workflow, the fresh produce can go bad, making it difficult to handle. This is one reason why food pantries keep and distribute a disproportionate amount of canned and packaged goods. They're easier to handle.
Our proposed solution is Produce Mobile and Kai Fresh. Produce Mobile is a mobile storefront for fresh produce. Donated and inexpensive fresh produce would be loaded on the Produce Mobile trucks and driven to areas in the Big Bend where our neighbors are experiencing food insecurity. Fresh produce in food pantries, food banks, and on the Produce Mobile trucks would be treated with Kai spray, a chitosan-based protective coating that extends the shelf life of the food. Finally, the fresh produce would be wrapped in Kai wrap, a chitin-based bioplastic that is biodegradable. Produce Mobile is expected to increase the availability of fresh produce to food insecure communities. The Kai Fresh system is expected to increase the shelf life of fresh produce by 2 to 5 days, increasing access to balanced meals to as many members of our community as possible.
With Produce Mobile, we are proposing to sell fresh produce in food deserts with a mobile food truck to increase access to nutritious, affordable food.
Justin Greer, Second Harvest, recommended locations where Produce Mobile could be located within communities in Tallahassee that experience food insecurity. The Produce Mobile business model was developed with guidance from Mark McNeese, a local social entrepreneur. The existing solutions have shortcomings. Farmers markets can help with food insecurity but are only available once a week and can be expensive. Farmers markets like the Frenchtown Farmers Markets work with Feeding Florida to decrease prices, but the low frequency of events remains an obstacle. Corner stores do not offer a variety of fresh food, and grocery stores can be too far away from the communities experiencing food insecurity, as there are not too many options for high nutritious food. These communities are known as food deserts, which are defined as areas where citizens live 1 to 3 miles away from grocery stores. Produce Mobile fills the gap at the intersection of grocery stores, farmers markets, food swamps, and food deserts. We talked to Second Harvest of the Big Bend to create a marketing plan by offering coupons to gain community buy-in, identified a pre-sales and scheduling system to help the community shop at Produce Mobile. Our business model indicates that Produce Mobile's operating costs would be around $60,000 per month. Expected revenue would be $65,000 per month. Produce Mobile has the potential of being a self-sustainable business with gross margins of 10%.
Chitin serves as the main structural material of arthropods and fungi. It can be found in many species in multiple kingdoms for a variety of uses. Chitin is based on glucose, the central energy storage molecule used by life. Deacetylation of chitin produces chitosan. Chitosan has antimicrobial properties. Chitosan is slightly positively charged due to the amine group. The cell wall and membrane is slightly negatively charged. The chitosan is attracted to the cell wall and disrupts the membrane. To determine if our solution would have a meaningful impact for our stakeholders, we bought green beans and tomatoes from Publix and placed them in a large box. In addition to our chitosan spray, we mixed in cinnamon and chokeberry oils that have antimicrobial and antifungal properties, to see if they would improve our solution's ability to preserve fresh produce. After washing and drying the vegetables with diluted water, we sorted the tomatoes and green beans into 4 equal groups. We then tested the preservative effect of our market-available chitosan foliar spray, High Shield, on the tomatoes and green beans. Using one group as control, we then dipped the 3 treatment groups into High Shield, a High Shield chokeberry mix, and a High Shield cinnamon oil mix. We found that the treatment group exposed to the High Shield and cinnamon oil mix had an extended shelf life of 3 to 4 days longer than our control. When we add an acetyl group to the amine group, we get chitin. While chitosan has chemically preservative properties, chitin has desirable structural properties, as a carbonyl group forms hydrogen bonds with the amine group to form a tight net. These structural properties allow the chitin to form a layer that has similar structural properties to petroleum plastics. Chitin, having evolved so early, is non-toxic to most organisms because they evolved a way to handle or digest it. It's recorded to be as toxic as raw glucose and can be consumed with no adverse effects. You have probably ingested chitin if you have eaten mushrooms, insects, or drank beer. Chitin and chitosan being so abundant serves as a major carbon source for many ecosystems. This is why chitin is one of the easiest polymers to degrade. The proposed Chi-Fresh system highlights the potential of chitin and chitosan to extend the shelf life of fresh produce. Success in extending the shelf life of fresh produce would be helpful to communities experiencing food insecurity. It also has the potential to impact all consumers. Longer shelf life contributes to making fresh produce more readily available to everyone. The full potential of chitin and chitosan is limited by the availability of the material. Chitin is sourced mainly from shellfish, which is expensive and unsustainable. Purification of chitin from shellfish requires a highly alkaline solution that then needs to be neutralized, which adds to the cost of chitin. We propose that microbial production of chitin should eventually be less expensive than sourcing chitin from shellfish. We pursued microbial production of chitin by designing, building, and testing the chitin secretion cells, which are engineered E. coli intended to secrete chitin. The chitin produced by the chitin secretion cells is expected to be easier to purify, less expensive, scalable, and sustainable. We also pursued upcycling brewer spent yeast, also known as BSY. BSY is a potential source of chitin, and it can be converted into a medium for growing the chitin secretion cells. The chitin secretion cells were designed to secrete chitin. The cells contain the NOD-CIJ generator, a genetic device designed to express the proteins NOD-C, NOD-I, and NOD-J. NOD-C is an N-acetylglucosaminotransferase that polymerizes UDP acetylglucosamine into chitoligomers with 4 to 6 residues, which is a kind of chitin. NOD-C removes UDP from UDP acetylglucosamine and covalently bonds the growing chain of acetylglucosamines. NOD-I and NOD-J are thought to assemble into a complex that is able to transport lipocytooligomers across the cell membranes. There is experimental evidence that the NOD-I-J complex can transport cytooligomers. We were unable to find structural models of NOD-C, I, and J. The NOD-C model used in the video is an N-acetylglucosaminotransferase from Staphylococcus aureus. As part of the modeling work, we decided to use AlphaFold, a machine learning system that predicts protein structures using DNA sequences, as the input. The models of NOD-I and NOD-J shown in the video are AlphaFold predictions that have not been verified experimentally. 3 versions of the NOD-CIJ generator were designed, each with a constitutive promoter of increasing strength. They were called NOD-CIJ generators 1, 2, and 3, with weak, medium, and strong promoters respectively. The NOD-CIJ generators were assembled and inserted into PSB1K3 plasmid backbones in a 1-pot, scarless assembly using the NEB Builder Hi-Fi DNA assembly kit. The assembled plasmids were used to transform E. coli NEB5-alpha. Only the NOD-CIJ generator 1 resulted in transformants which we called the chitin secretion cells. The chitin secretion cells were grown in LB medium overnight, centrifuged, and the supernatant was collected. The supernatant was evaporated in a chemical hood overnight to increase the concentration of any chitin that was in the supernatant. The residual material was resuspended in deionized water to a concentration that was 12 times the original concentration. An equal volume of 12.5 m NaOH was added to the supernatant and the samples were placed overnight in a 95 Celsius water bath, which converts chitin to chitosan. The chitosan assay kit from Cell Biolabs was used to detect chitosan in the supernatant. The chitosan assay kit provides a colorimetric readout of the chitosan concentration. The results can be quantified by measuring in a microplate reader the absorbance by the samples of 540 nanometer light. The results of our test are summarized on the graph. The horizontal axis lists the samples and the controls. The first column is a supernatant of the chitin secretion cells. The second column is LB medium that was tested with the chitosan assay kit. The third column is deionized water that was tested with the chitosan assay kit. The fourth column is LB medium that was not tested with the chitosan assay kit. The vertical axis is estimated chitosan concentration in micrograms per milliliters. The estimated chitosan concentration in the supernatant of the chitin secretion cells and the tested LB medium were 256 and 217 micrograms per milliliter, which was not significant. The results indicate that there is chitosan in LB medium, and it is undetermined if the chitin secretion cells secreted chitin into the culture medium. We will be repeating the measurement after growing the cells in M9 minimal medium. We will report the results during the jamboree. To reduce the cost of microbially produced chitin, we looked at a new way to source chitin: brewer spent yeast. By upcycling brewer spent yeast, we'd provide a more sustainable, scalable way of sourcing chitin. Brewer spent yeast is the yeast left over from the production of beer. 30 million tons of brewer spent yeast is produced every year, but most of it is used for feeding livestock or sent to a landfill. With brewer spent yeast, provided to us by Proof Brewery, we are evaluating if brewer spent yeast can serve as an inexpensive growth medium for the chitin secretion cells. In partnership with the University of Pennsylvania iGEM team, we are characterizing 2 proteins identified in the UniProt database that may be chitin synthases. If successful, these chitin synthases would improve the microbial production of chitin. To accomplish this, the UPenn iGEM team are utilizing their OptoReader to test the light-inducible expression of these chitin synthases. Greater control of our cells' production of chitin would enable us to prevent chitosan from killing our cultures, since chitosan is toxic to the cell. We partnered with the Lambert High School iGEM team in Sewanee, Georgia to further reduce the cost of microbe-produced chitin. We shipped an agar plate of chitin secretion cells to Lambert. Lambert freeze-dried the cells using LiFo-X, an innovative and inexpensive lyophilizer. Lambert sent the lyophilized cells to the Johns Hopkins iGEM team to test that the cells could be reanimated. If successful, we expect that Lambert's lyophilizer will lower the cost of storing and shipping chitin secretion cells. For the bronze medal requirements, we produced a wiki, the presentation video, the judging form, and we curated a collection of biofab promoters from the registry as a contribution to the iGEM community. For the silver medal requirement, we collaborated with the UPenn, UF, GHU, MIT, and Lambert iGEM teams, collectively known as the Eastern Seaboard Alliance. We spent months troubleshooting our projects over Zoom. We analyzed the impact food insecurity has on health, consulted experts on the impacts of the project, documented the parts of our solution, and proposed an implementation plan for our project. For the gold medal requirement, we partnered with the Lambert High School team to reduce the cost of storing our chitin secretion cells, integrated our stakeholders' feedback into the design and implementation of our project, modeled the microbial production of chitin, the AlphaFold non-IJ complex, and a business report of our KaiFresh startup.